Okay, in the second video, we are going to discuss clinical progression from being HIV positive into full-blown AIDS, and then we'll go through molecular virology, basically walk through the replication cycle of HIV, some of which we have seen before. So in terms of clinical progression, in this graph, you are looking at the number of helper T cells, CD4 T cells per microliter of blood. And of course, this is because these are the cells that HIV preferentially infects and that become depleted as you approach AIDS and become immunocompromised. So we have the viral load being shown here as the red line, and then basically what's happening with the CD4 T cells shown as the blue line. So you have this acute phase that can last 2 to 8 weeks. We'll talk about that here in just a second. At the end of that acute phase, individual has undergone seroconversion, just meaning they have produced antibodies against the HIV virus. You get this initial spike in viral load, but then again at the end of that acute phase it is returned to a very low baseline and will stay there for potentially many years, right? 8, 9, 10 years. And this would be in the asymptomatic or a latency phase. So at this point the person has no symptoms, but their population of CD4 T cells, those important immune cells, are slowly but steadily declining, right? And so as we approach the end of that latency phase, then we start getting into a period where you've got an increased risk of secondary opportunistic infections. And really that point begins once the number of CD4 T cells falls below 500 cells per microliter. And once it drops below 200 cells per microliter, then somebody is considered to be in full-blown AIDS at that point, right? And that can last for 1 to 3 years. And so we'll come back. I'll go through each of these briefly here in a little bit more detail. So in that first acute stage, which as I said could last anywhere between 2 to 8 weeks, and usually this stage would begin anywhere between 2 to 4 weeks after the person was initially infected. You get typically pretty severe flu-like symptoms. So, all stuff that we're familiar with. And as we saw in that graph, you get a spike in the viral load. At the end of this period, you get the seroconversion happening. But it's very likely somebody may not know they've been infected with HIV, right? They probably just think, oh, I got the flu, I've got all these flu-like symptoms, feel poorly for a few weeks, and then recover and are back to normal. At the end of that, then we're into the asymptomatic or that latency phase, where, as I mentioned, right, there are no symptoms or maybe very limited symptoms. And at this point, HIV is just very slowly going through its replication cycle and infecting and killing CD4 helper T cells, shown here. So you may remember those from our immunology chapter. And this asymptomatic phase can last on average 10 years without any kind of anti-retroviral therapy. With appropriate combination drug therapy, we seem to be at the point now where we can extend that 10 years apparently indefinitely, right? We've had people that have been HIV positive for many, many years and have never moved out of this particular phase. But there's, of course, a high cost, right? A high financial cost associated with maintaining that therapy. In terms of this 10-year number, there are 2 notable exceptions here. So there are some individuals that were considered rapid progressors that could go through that average 10-year period in 2 to 3 years, so progressing to AIDS very quickly. And then on the flip side, you have long-term non-progressors, LTNPs, that will keep basically a normal CD4 T cell count and pretty low HIV viral load even in the absence of antiviral therapy. And so some of these long-term non-progressors are known as elite controllers, and they may even have no detectable virus by most common detection methods. And we'll talk about these non-progressors and elite controllers when we read our HIV book. It will feature prominently in that. So I already mentioned this, I think, right? But a normal healthy individual would have anywhere between 500 and 1500 CD4 T cells per microliter of blood. And if you're not familiar with microliters, there would be 1,000 microliters in 1 milliliter. So that's a very, very small amount. So you have a pretty large number of T cells packed into a very small volume. But of course that number is falling, right? We fall below 500 and we start to open ourselves up to other secondary infections. We drop below 200 and then we are in immunodeficiency full blown at that point. And of course this is almost always life-threatening, right? So a person that is in AIDS is typically going to live about 3 years on average, and maybe only a year after acquiring some other opportunistic infection, because the immune system is depleted to the point that even these what would normally be benign secondary infections are going to run rampant and become life-threatening. Okay, so hopefully that gives you a decent overview of the clinical progression. So as I said in this last section, then I want to go through the molecular virology, which is, of course, somewhat complex for HIV being a retrovirus. And we have seen pieces of this before, so some of the things that I have gone over I won't dwell on again. But just to set the stage in terms of the molecular structure again, right? Remember this is an enveloped retrovirus, so you have this kind of bullet-shaped capsid, and then you have the viral envelope around that. You've got this single capsid protein shown in blue here, and interestingly, the capsid contains 2 copies of the RNA genome, and remember this is a positive sense single-stranded RNA genome. So it's considered to be pseudo-diploid, sort of diploid. But I said it's interesting because really, apparently the virus really only needs 1 copy. Only 1 copy seems to be used for reverse transcription and going through the viral life cycle, so it's a little unclear exactly why it carries 2 copies, but it does. And if we look inside the capsid here, we'll see we have the viral RNA associated with the nucleocapsid protein, these dark circles. And then we have 3 enzymes, proteins there inside the capsid as well. We've got the reverse transcriptase here in yellow, as you can see is attached to the end of the ribonucleoprotein complex. And then you've got the protease, which we'll see is important for maturation, and an integrase, which is a key part of integrating into the host chromosome. And then we have the matrix protein, right? You always find a matrix protein in envelope viruses, so just on the inner surface of the viral envelope. And we have these glycoproteins then protruding outward on the surface out of the viral envelope that are trimers, meaning they're made of 3 pieces. And those 3 pieces are composed of these 2 different proteins, so GP120, glycoprotein 120, glycoprotein 41, which if you look at the cartoon here, the glycoprotein 120, like there's 3 of those subunits basically at the tip of that structure, that surface structure, and then the GP41 makes up the stock. And we'll see those, we'll talk a little bit more about these when we get to attachment here in a few minutes. Alright, so I'm going to go through the replication cycle piece by piece, but first I just wanted to kind of give you the lay of the land, give you like the quick run through of big picture, right? So keep in mind this cycle is preferentially happening in those CD4 T cells. So we have attachment, right, to the cell surface receptor and co-receptor. We get uncoating at the plasma membrane, penetration penetration at the plasma membrane, basically releasing the capsid into the cytoplasm. That capsid then breaks apart, uncoats there in the cytoplasm, releasing the viral RNA, which is then reverse transcribed into a DNA copy. The DNA copy goes into the nucleus where it integrates into the host chromosome, and then at some point, or at least at some steady low level, we do get transcription of the viral RNAs, right? And those get processed as would any other messenger RNA. They would leave the nucleus to go be translated, right, in the cytoplasm. So we're actually producing new HIV proteins. And then at this point, and of course then in the background, we're also having to make copies of the viral RNA, right? We'll come back to that. But once we've got the viral proteins, copies of the viral RNA, we can start to assemble new virions, and they're going to assemble at the plasma membrane, at the cell surface. They will be released through this process of budding, and then I think we mentioned this in previous chapters, but HIV is kind of unusual in that it matures after it has already budded out of the host cell, and then that virion would go infect another CD4 T cell. And so just to zoom in on the beginning of that process, here you can see a single T cell basically in blue. All these little yellow specks here are HIV virions attaching to the surface. So you can see this T cell is really under attack, right? It's got a ton of virions attached to its surface. And if we think about, okay, well, how does HIV get to the T cells, right? Because primarily, it's it might be acquired through sexual intercourse or through a blood transfusion or some of those other methods that we already talked about. Well, however it initially enters, we get HIV in one or more tissues in our body, and if you remember back to our immunology chapter, you have dendritic cells and macrophages, right, basically kind of scouring all tissues looking for foreign invaders, and in some instances HIV can infect certain macrophages, but the primary route is thought to be it will attach to dendritic cells by binding to this DC sign receptor protein, and then ends up inside the dendritic cell. And keep in mind, dendritic cells then travel back to lymph nodes where they're presenting antigens that they found to those helper T cells. And so in a nutshell, what will happen is when the dendritic cell gets back to the lymph node, HIV virions can escape from that dendritic cell, and then of course their favorite target, the CD4 T cells, are right there. So essentially I want you to think of HIV is the fact that HIV is using these dendritic cells as basically like a Trojan horse, right? It's hiding inside of them and popping out at the last minute to attack. All right, so let's zoom in on the attachment. How does HIV attach to a helper T cell? And so we've already seen these receptors before. You have the primary receptor CD4 and then one of two co-receptors. Mostly we focus on CCR5, the CXCR4. It's usually you only tend to find that later in the infection process or in very newly differentiated T cells. But as we'll see in our HIV book, we read the CCR5 co-receptor kind of features prominently in this whole process. But what researchers have found is it goes something like this, where the glycoprotein 120, right, which is the trimer that's at the tip of these structures on the surface of the virion, will bind initially to CD4, causing it to kind of bend over, get this conformational change, and then that kind of allows the glycoproteins to interact with the co-receptor, So we get this binding to the co-receptor, and when that happens, it will actually reveal this what's called a fusion peptide within the GP41, which is the stock portion of those structures. So we get this little conformational change, and basically that a portion of that GP41 is able to insert into the plasma membrane. Okay, and then it will refold such that it creates a pore in the plasma membrane. So we're having attachment and penetration basically occurring together here, and then that's going to allow that capsid core to be passed across the plasma membrane into the cytoplasm. And so in terms of uncoding, we don't really know much about it, that process. We know it happens apparently in the cytoplasm, but there has been proven to be difficult to study. And then that leaves us with the next step would be, okay, after we're uncoded and we have the viral RNA out, we need to reverse transcribe, right? We want to make a DNA copy of our RNA genome. And I won't go through all these steps because I went through them in chapter 4 in some detail, but just keep in mind, right, the reverse transcriptase enzyme that carries out this process is already there. It's attached to the viral RNA. It was loaded in the capsid. And also keep in mind that the reverse transcriptase has a few different functions, right? So it can make DNA from an RNA template, an RNA-dependent DNA polymerase. It can make DNA from a DNA template, and then it can also degrade RNA that's part of these RNA-DNA duplexes. And it doesn't proofread, so it will make mistakes. And also keep in mind this whole process utilizes this tRNA primer, which actually was stolen from a previous host cell, but it comes kind of preloaded on the viral RNA. And just keep in mind a lot of polymerases, right, need that 3' end of a primer in order to start being able to build new DNA. Same kind of story here. And so we go through this, this kind of elaborate dance of steps where you've got, pieces jumping from one end of the nucleic acid to the other. In this cartoon, right, the white is the original viral RNA and everything in green is new DNA that's being synthesized. But through these steps and the activity of the reverse transcriptase, we end up with a complete double-stranded DNA copy of our viral RNA. Okay, and like I said, we already went through that in chapter 4, so I'll refer you back to that if you're having trouble working through this here. Most retroviruses, they need to get into the nucleus after that reverse transcription. They want to get the DNA in, and they'll typically have to wait until the cell undergoes mitosis and the nuclear envelope starts to break down in order to be able to get access to the genome. But the HIV complementary DNA is actually able to pass straight through the nuclear pore, so it can go straight in. And you get what's called the pre-integration complex, which is basically the beginning of that integration, and you have the integrase enzyme, right? Remember, which was one of those enzymes loaded in the capsid. Again, we already kind of went through this, but that will facilitate that process of integrating the viral DNA into the host cell chromosome. And it's not believed to occur at any specific directed site, it just seems to happen basically at random. Although it does usually seem to happen in areas that are more transcriptionally active, right? So it's not happening like at the telomeres or in the centromere of the chromosome, which are places where you really don't find many genes and don't get much transcription happening. So it does seem in some way to be targeted to transcriptionally active portions of the chromosome, but other than that, there doesn't really seem to be any preference for specific sites. Then once we're integrated, we have to replicate, right? So, and express the genes that are encoded in the genome. So the HIV viral DNA has 9 open reading frames, which is basically just a fancy way of saying 9 genes, but those 9 genes actually code for 15 proteins. And how could that happen? Well, we could do that through this phenomenon of alternative splicing, or in some cases, one of those genes may actually encode one of these, like sort of polyprotein precursors that can then be chopped up by proteases into multiple active proteins. So don't worry too much about the details of this, but it's just kind of showing you the layout of where these genes are in the integrated HIV genome. And so then we'll use like host transcription factors. They're going to bind to usually the long terminal repeats, these LTRs at the end, and that's what's going to attract RNA polymerase and allow for transcription of these genes to happen. Usually the very first genes to be expressed are this TAT and REV, transactivator of transcription, regulator of expression of viral proteins. So these are going to produce proteins that are really going to get the ball rolling and ramp up transcription and ensuing translation of the viral genome. This REV also has this job of taking unspliced transcripts from the nucleus out into the cytoplasm, which is kind of unusual because usually if you have any messenger RNAs that were supposed to be spliced but weren't, they get kind of retained in the nucleus until that splicing occurs. So that was these genes, these proteins here. As I alluded to, there are actually 3 of these polyproteins that will get translated and then spliced by proteases into individual proteins. So you get what's called the GAG precursor down here. This one is not going to end up being spliced until after the virion has already been released. So that's part of that maturation process, and I'll come back to that. I think I may have mentioned that previously in another chapter as well. Then you also have the GAG-Pol precursor shown down here, and this only happens about 5% of the time, so this will be due to the ribosome just frame shifting, some low occurrence, but if that's the case, that will then need to be cleaved into these 3 individual proteins. And then lastly, we have the ENV precursor shown over here, which is going to produce those 2 glycoprotein subunits, right, that we're using at the cell surface for attachment. And then all the other proteins are just being produced as sort of traditional, full-length messenger RNAs that may or may not be spliced, but through a more normal route that cellular genes would be transcribed and translated. And so then I won't delve too deep into replication here, but just to remind you, right, we've got our viral double-stranded DNA, and we need to make RNA copies, right, those positive sense RNA copies that could be packaged back up into new virions. So we use RNA polymerase II for that, and those RNA copies will bind to a cellular tRNA that will be kind of preloaded as that primer for reverse transcriptase, right, in the next infection cycle, next replication cycle. And so then that gets us down to assembly, release, maturation. And as I showed you in that initial replication cycle, so assembly is happening at the plasma membrane, right? So some of these viral proteins will actually embed in specific regions of the plasma membrane, accumulate. Those will then attract the viral RNA, and it will begin to bud off, eventually fully budding out and surrounding the viral RNA. But keep in mind that the initially budded virus is still immature, so you have that the GAG-Pol precursor polyprotein that has to be cleaved into 3 different proteins in order to create that truly infectious virion and the fully formed capsid that we saw in the beginning. And so yeah, that's it. I will stop there. But yeah, there's a lot going on with HIV, but I think it's a particularly interesting one. As we discussed, it's still very, very medically significant worldwide, so definitely worth talking about.